Now let us learn about the steps in genetically modifying an organism in detail. So first is identification of a DNA with a desirable gene. Traditional techniques like hybridization, you take two organisms and hybridize them with a desirable quality. It does not mean that the hybrid will give rise to the hybrid will have the desirable quality only. It will also include the undesirable genes when it is getting crossed. So traditional hybridization techniques leads to inclusion and multiplication of undesirable genes and desirable genes. But in genetic engineering, only the desirable genes are introduced. Only the DNA with the desirable gene is identified and introduced. The next step is introduction of the identified DNA into a host. A vector DNA such as plasmid is used to deliver an alien piece of DNA into a host organism. Suppose we know what are plasmids. They are single-stranded, self-replicating, extra-chromosomal DNA which is present in prokaryotes. So a plasmid is a very good vector, and this vector is very useful in transferring a gene of interest, that is an alien piece of DNA, from a recipient into a host. So a vector DNA such as plasmid is used to deliver an alien piece of DNA into a host organism. You can see, suppose this E. coli is the host, and this is the plasmid. So into this plasmid, we insert the gene of interest. So this is a recombinant plasmid which has the gene of interest, a plasmid which is carrying a gene of interest, and that it is introduced into a host organism. So this is a method. This is the second step. There's introduction of an identified DNA into the host. The third step is maintenance of the introduced DNA in the host and transfer of the DNA to its progeny. So we have learned in DNA replication, where does the replication of DNA begin? It is called as the ORI site, or it is called as the origin of replication. And this alien piece of DNA, it cannot multiply itself in the progeny cells of the organism because it does not have an ORI site. Hence, the alien DNA is integrated into the recipient genome or the chromosomal DNA, which is a part or which is having an ORI site. So an alien piece of DNA which is linked to the ORI site or origin of replication will undergo replication and multiplication along with the host genome. So that's the third step, maintenance of the introduced DNA in the host and transfer of its DNA to its origin only if it is linked to the ORI site of the host genome. The process of joining and inserting a foreign piece of DNA into a host organism to produce new genetic combination is called as recombinant DNA technology. It is nothing but mix and match. Mix and match is nothing but biotechnology. So a process of joining and inserting a foreign piece of DNA into a host. So a DNA of interest is removed from another organism and it is introduced into a new organism that is a host organism to produce a new genetic combination is called as recombinant DNA technology. And the first recombinant DNA or rDNA, recombinant DNA, was produced by Stanley Cohen and Herbert Boyer. So these are the two scientists who produced the first rDNA, Stanley Cohen and Herbert Boyer. What do? They isolated an antibiotic resistant gene, a gene which is able to give an antibiotic resistance. And it was isolated from a plasmid of Salmonella typhimurium. So a gene for antibiotic resistance was isolated from a plasmid of Salmonella typhimurium. It's a bacteria. And it was linked with the plasmid, that is a vector. Okay, the gene was isolated from Salmonella typhimurium and it was linked into a vector. And with the help of the vector, it was transferred into E. coli. So E. coli is the host organism. As a result, the gene was expressed and multiplied in E. coli, that is the host organism. So this was the first rDNA which was done by Stanley Cohen and Herbert Boyer. So dear children, in this module, we learned about the steps in developing a genetically modified organism and the first recombinant DNA which was developed. Thank you.